Now it's time to look at my personal favorite of the macromolecules, proteins. Why are they my favorites? Well, to put it simply, they're pretty much the superheroes of the macromolecule world. They have a multitude of different purposes and are key for life to take place. They serve as support for the immune system, structural support for cells, they're enzymes, they help transport molecules around the bodies of living organisms, and just so much more. So before we dig into the meat of proteins, which is kind of funny because meat have high concentration of proteins, we should look at their structure. Now, this is kind of difficult because proteins have extremely complicated structure. So what are proteins made of? Well, their monomers are amino acids. Their polymers are called polypeptides. But a polypeptide by itself is not a protein, and we'll get into that later. Their polymers are, sorry, I already said that. To understand the protein structure fully, we need to dive a little deeper into amino acids. Amino acids are constructed of a carboxyl group, an amino group, and most importantly, an R group or side chain. These side chains are what makes each individual kind of protein unique, because these side chains make each individual amino acid unique. And thus, depending on the sequence of amino acids in the protein, you're going to have a different sequence of R chains in the protein. Now that we know that, let's look at the polymers, polypeptides. Polypeptides are long chains of amino acids held together by peptide bonds. These bonds are formed by dehydration synthesis, what we talked about earlier, where two monomers or two polymers, or monomer and polymer, will form a water molecule, and that water molecule will separate from them, and they will have a bond left over holding them together. So after this point, protein structure gets a little complicated because it branches off into 4 different levels. The first level is just the polypeptide chain. The second level is the coiling and folding of these polypeptide chains. The third level is caused by the various chemical interactions between the side chains of the amino acids. And the fourth level of structure is the result of 2 or more polypeptide chains combining. When this happens, when you get to the fourth level of structure, each different kind of protein has a unique shape, a unique 3D shape that's extremely complicated. It's often very coily and almost kind of messy looking. So, now that we have taken a look at protein structure, let's look at some different kinds of proteins. Number 1, enzymes. Enzymes are really interesting. They are biological catalysts. A catalyst is something that speeds up chemical interactions between substances. So, enzymes speed up these biochemical reactions, right? These enzymes, but we need to know how they do it and why they do it. So, the way it happens is you have your enzyme, which I'm going to draw a very simple shape to represent this. We're going to do a square with a little cutout in it. Well, more like a rectangle with a cutout. We're going to label it E for enzyme. There is our enzyme. So, enzymes bind to substrates, which are specifically shaped to bind to them. Now, this sounds really, really confusing, but once you see a diagram, it's much less confusing. So I'm going to draw us a substrate and label it S for substrate. So that little tiny rectangle there is our substrate. So that substrate is going to bind in the enzyme, and this area in here called the active site. So when they bind together, sorry, I got a little bit of marker on my finger. The enzyme will either break down the substrate into maybe 2 or more different pieces, or if it has multiple spots for binding, which a lot tend to, it will sometimes bind substrates together to form, or sorry, they don't have multiple different spots, but one spot can host multiple different substrates. And when they do this, they'll bind them together and create a new substance. So earlier we talked about cellulose and how we don't have the enzymes to digest it. Now that should make more sense, because if we don't have the enzyme to digest it, it's just going to sit around in our body for a really long time. We might be able to digest it on our own, but it would take super long amounts of time because enzymes speed up these reactions. This is why enzymes are significant and needed for life. They make biochemical reactions inside of living organisms happen in reasonable amounts of time. Without them, things would take forever to happen, and people just wouldn't be able to live. So we're going to look at our next kind of protein, which is structural proteins. So proteins are used to build up muscles and tissues. These kinds are structural proteins. You've probably heard about bodybuilders who ate insanely high amount of protein, right? Like to build up their muscles, to get bigger. Well, this is the principle behind it. These proteins are used to build the muscles and tissues inside of you. For the same reason, growing children should also eat a good amount of protein as their body tries to build muscles and tissues in them. This is why teenagers often eat a lot, especially during the years that they're having their growth spurt. Now, there are tons of other variety of proteins, but these two are really important to know. Now, there are a couple other tidbits about proteins that we should definitely keep in mind. Number one, remember the polymer of proteins, polypeptides? Well, as I mentioned earlier, a polypeptide itself is not a protein. Proteins are often multiple polypeptides that have been formed into a shape unique to them, like I talked about earlier, that unique 3D shape. Number two, that's one of the other important things. Each kind of protein has a highly unique 3D shape. And number three, that also is connected to numbers 1 and 2, most proteins, if not all proteins, rely on their shape to do their job. Take an enzyme, for instance. If it loses its shape, this is called being denatured, usually due to being exposed to temperatures that it's not built to handle, so either like highly cold or highly warm temperatures, or if it's exposed to a pH level that it's not built to be exposed to. What'll happen is the enzyme will lose its shape. So take our rectangle guy with the cutout. If he got denatured, he might start like bending in the middle or something, just a simple example. And then I'm going to label him DE for denatured enzyme. That substrate isn't going to fit in there anymore. So he can't catalyze this reaction, and he's no longer biologically useful. So a protein loses its shape, it most likely can't do its job. Occasionally a protein will be renatured after being denatured, but this doesn't always happen. It can't always happen for certain enzymes and proteins, but sometimes occasionally it'll take place. So now it's time to look at the fourth and final macromolecule group, nucleic acids. Now you're probably like,